Good afternoon. My name is Audrey Guyonnet. I'm a member of the Guyonnet Lab, and I study how artificial selection can alter brain morphology. For over 30,000 years, humans have artificially selected animals for a diverse number of traits. Most often, these are conducive to commodity production, like food and leather products. However, we have also selected these animals for behavioral traits as well, like navigational ability. So it's possible that selecting for these behaviors has influenced the morphology of the brain. In neuroscience, this is best explained by the principle of proper mass, wherein a brain region increases in size relative to the animal's ability to perform the behavior that that region controls. In domestic animals, this is the theory behind the regression hypothesis, wherein domestic animals have shown a decrease in brain size in comparison to their wild ancestors. This is thought to be due to the simplicity of captive living. So without threats of predation and interspecific competition, more complex behaviors like predator evasion are no longer necessary for survival. Therefore, the brain could have reduced in size as these behaviors have become less expressed. But what about behaviors that have been augmented by selection? For my research, the rock pigeon works as a remarkable model species. So over the last 8,000 years, these birds have been selected for an incredible array of features, with over 350 breeds distinct in their plumage, size, and flight style. Pigeon breeds can be classified into one of three groups based on function: the exhibition breeds selected for plumage or morphological characters, the utility breeds for meat production, and the sporting breeds who specialize in athletics like long-distance racing, high-altitude flight, or aerial performance. Perhaps the most famous of the sporting breeds is the homing pigeon, which has been used to deliver messages since 3000 BC. So this relies on the incredible ability of pigeons to home, an innate navigational behavior used to locate their nest after daily foraging sessions. They do so by combining external cues to create a map-like representation of space. For wild rock pigeons, homing was useful for locating a cliffside nest from up to 80 kilometers away with very few visual cues, like across an ocean. Today, the domestic homing pigeon has been selected to locate a home loft from a novel starting point over unfamiliar terrain from up to 1,000 kilometers away. Like other navigational behaviors, homing is controlled by the hippocampus, and I believe that selection for homing has placed greater demands on this brain region. Because of this, I predict that homing pigeons will have larger hippocampal regions with more neurons compared to non-homing breeds in order to accommodate these increased behavioral demands. To estimate the volume of the hippocampus, I used Stereo Investigator software to calculate total brain and regional volume. Here, I compare relative hippocampus size on the y-axis, using the telencephalon as a scaling structure on the x-axis. Because breeds differ in housing style and, as a result, body conditioning, using a regional scaling structure can be more accurate than using something like relative body mass. By performing an ANOVA in R with a Tukey post-hoc test, I was able to show a very significant difference between the size of the homing pigeon hippocampus and all other breeds. And you can see that the y-intercept of hippocampal volume is much higher in homing pigeons, shown in orange, than other breeds we have studied so far. To estimate the number of neurons in the hippocampus, I fluorescently labeled neuronal nuclei, as shown in the image below, and counted them using Stereo Investigator. Here, I compare neuronal populations between pigeon breeds. As you can see, the feral and sporting pigeons show significantly lower numbers of neurons within the hippocampus than the homing pigeons, decreasing by anywhere between 45 and 62%. The breeds we have used for comparison have so far been active breeds, which is interesting because exercise can increase expression within the hippocampus. We may therefore see an even more dramatic decrease in neuron number within the exhibition breeds, who are more sedentary in lifestyle. So to summarize, my preliminary results have shown that intensive artificial selection for behaviors can in fact alter a brain region. And the comparison of homing to feral pigeons is especially interesting, given that feral populations are made up entirely of escaped homing pigeons once used to deliver messages across North America. Such a dramatic decrease in brain region size over such a short period of time is a testament to how quickly relaxing those same selective pressures can also alter the brain. Next, I hope to add exhibition breeds to this comparison and to compare flight styles of different active breeds within our aviary. Thank you for listening, and thank you to NSERC, CFI, and the University of Lethbridge for making this research possible.
Thank you, Audrey. That was really interesting. So if there are any questions, please type them in the Q&A box. So you found that selecting for behavior affected the makeup of the brain, specifically the hippocampus. And I'm just wondering, do you think that this would also be the case in, well, I'll just say it in chickens or really any kind of domestic poultry, as there is some selection for certain types of desired behaviors. It's more for production traits, obviously, but I'm just wondering if you think on the behavior side, it might apply to commercial poultry as well.
Yeah, for sure. So a lot of the work in this area has looked at stress responses and fear responses in chickens. So when you're selecting for that tameness factor, you're selecting for a decrease in fear responses. So I know Per Jensen works out of Sweden and studies the domestication of the domestic chicken. And so they've been able to show that brain regions have decreased in size, specifically the telencephalon has decreased in size over the course of that domestication.
Wonderful. Um, so I'm also wondering, having never worked with pigeons or pigeon brains, how did you find that? Was it difficult?
It was a very new experience to me since my background is not in neuroscience, but I've done a lot of animal behavior work. So this has been really interesting, and I had no idea that pigeons could be so diverse and especially when we look at their brains, despite being able to show that there's a difference in volume, which is what we're interested in. If you look at the brain itself, you can tell whether you're looking at a homing pigeon versus a regular or exhibition breed, just based on the shape of the brain.
That's fascinating. Um, we have questions as well. Are there any, in the Q&A box, are there any negative consequences to increase in brain size?
So usually brain size would be constricted based on how you've selected the bird for its head shape. So that's something we'll see. I can't see any negative consequences to increasing the size because you're constrained by head shape and size. Probably increasing one region size could potentially decrease another brain region size. But so far that doesn't seem to be the case for our study.
And very good. Two more questions. Let's see if we can do them. Does it mean a big brain and birds become smart? So basically, are smart birds having big brains? What is significant to the domestic birds for farming?
So sorry, is the question what is significant about a bigger brain?
Yeah, does it mean the birds are super smart?
Yeah, so that's basically what we're trying to show is that when you select for something like that, the bird is becoming much smarter. So in this case, when you're looking at volume, we're not entirely sure. It could be an increase in the number of neurons or an increase in the size of the neurons. But anytime you increase complexity of neuroanatomy, you're going to be increasing the cognitive capacity of that animal.
Fantastic. And last one, will you plan to compare their RNA or protein expression?
Not currently. I know that my advisor is very interested in continuing this work in domestication. So as soon as we receive some birds in our aviary, we're going to be looking at doing genetic testing as well. One thing we're very interested in is looking at, it only takes a few breedings between different breeds of pigeon to revert to those ancestral characters. So what you see in a wild pigeon. So we'd love to see the genetic factors at play there and what's combining to basically cause a reversion to ancestral characteristics.